Hello and welcome everyone. Over the next 2 days we're going to talk about various molecules that are important for life. As we begin that discussion, we're going to want to talk about some small molecular components of larger molecules, and we call these functional groups. These will be smaller chemical structures that we find on larger molecules that give them a function, as the name sort of implies here. We're going to talk about how large molecules are formed. And then we're going to spend the rest of this podcast and the following podcast talking about large macromolecules. And there are 4 macromolecules that are associated with living organisms. The first are carbohydrates. These are what we commonly call sugars. And then we'll talk about lipids. These include things like fats and steroids. Then we'll talk about proteins. And then we'll close with talking about nucleic acids. This would include DNA and RNA. In this podcast, and for day 6, we're going to talk about functional groups, how large molecules are formed and some terminologies associated with that, and carbohydrates. For day 7, we'll talk about lipids, proteins, and nucleic acids. So let's go ahead and get started. As we begin this discussion of these large macromolecules, it's important to know that they are made of various series of what we call hydrocarbons. Hydrocarbons, as the name implies here, are structures that are made out of the element hydrogen and carbon. As we begin this discussion of hydrocarbons, let's think about 4 ways that carbon structures can vary. That is the carbon skeleton, and how that can vary. The first is length. This simply just means that you may have a 2-carbon structure, or you may have a 3-carbon structure. These are both hydrocarbons, but they vary in length. Hydrocarbons could also vary based on whether or not they form branches. So what do I mean by branching? Well, let's draw a carbon skeleton where there's no branching first, like so. Now, you could also have a branch forming, say, from this carbon, where you add a carbon like so, maybe another carbon, just like that. So this would be an example of how the carbon skeleton branches. You can have multiple branches. You could have a branch coming off here, you could have a branch coming off here. They vary in the way they branch as well. Now I'm going to get rid of this bottom carbon branch here so I can talk about the third way that carbon backbones can vary. And they can vary based upon double bonds forming, as well as how many double bonds are present and where they are present. So you could have a carbon structure like this, where you have 4 carbons, each surrounded by hydrogen. I didn't mention it in the beginning, but it probably is worth making a comment on. Every carbon binds to 4 different elements. So this carbon here binds to hydrogen, and another carbon, and hydrogen. Now the only time that varies is if there is a double bond present. So if we look at this structure here that I've just drawn, we can see that there are single bonds connecting the hydrocarbon skeleton. And as such, each of these carbons has 4 different elements attached to it. However, if we make a double bond here, that removes this hydrogen. Or you could remove the top one, it doesn't matter which one. But you've reduced the ability of the carbon to bind to another element. So this would be a double bond. Now this last way that carbon backbones can vary is the presence of rings. And I did not leave myself much room on the bottom here, so I'm going to write it up top as opposed to trying to squish it into the bottom. And so I'm going to just write presence of rings. What we can see here in all of these cases, the carbon structure is linear. The carbon could form a ring like so, where each of these points is a carbon. So I'll just make a C at each of these. They also have hydrogens attached to them as well. These are the 4 ways that carbon skeletons can vary. And any given carbon structure could vary in one or all of these different ways. Next, I want to spend a little time talking about functional groups. Now, there are 7 functional groups that you need to know and be able to recognize. In order to memorize these, I would recommend maybe making flashcards. Probably the best way to memorize these. So let's first list the different functional groups. Hydroxyl groups, carbonyl groups, carboxyl groups, amino groups, sulfhydryl groups, phosphate groups, and methyl groups. So you should be able to list these, and you should be able to recognize an example. I'm not going to ask you to draw a hydroxyl group, but I very well may ask you to identify a hydroxyl group, a carbonyl group, an amino group, a methyl group on a given structure. So a hydroxyl group, let's draw that first. That is an OH. A carbonyl group is a structure that I'm going to draw a little bit to the right of the hydroxyl group just so we don't run into it, where you have a carbon, and then you have a double bond O, and then it is connected to 2 other carbon structures like so. Or it could be a hydrogen. A carboxyl group is similar to a carbonyl. It's similar in that you have a carbon group with a double bond oxygen on it, and it's also connected to either carbon or a hydrogen like so. And on this side, unlike the carbonyl group where it's a carbon group or a hydrogen next to it, it has an OH group. If you can remember what a hydroxyl group is, that helps remember what the carboxyl group is. Because both the hydroxyl group has an OH group, just like the carboxyl group has an OH group. And so this bottom part of the word here, last part of the word, is the same. So they both contain a hydroxyl group. An amino group is going to have a nitrogen and hydrogens attached to it. If you have a hard time remembering this, this may or may not help. Only amino and carbonyl have an N in the names of them. If you can remember carbonyl because it's similar to a carboxyl group, then that might help you remember that the only other one with an N in it is an amino group. The next 2 are a little easier to remember, I think, because the sulfhydryl group has a sulfur in it, like so. Just like the phosphate group has a phosphorus in it. And then finally the methyl group has the carbon attached to 3 hydrogens. You should be able to, as I said before, list these. If I gave you one or multiples of these, you should be able to tell me which one it is. We'll probably have a practice with a question like this in class, but certainly on the test I could see a question like this coming. I may give you a larger structure, and maybe this larger structure will have multiple functional groups. Maybe it'll have a carboxyl group here. Maybe it'll have a phosphate group. Maybe it'll have a sulfhydroxyl group here. Maybe it'll have a methyl group here. So any given carbon structure could have multiple functional groups, and you may be asked to identify all of the functional groups on a given structure. So be very familiar with these. One thing I will not ask you to do is ask you to draw a hydroxyl group or carbonyl group, carboxyl group, or any of these. You won't have to draw them. Okay, let's go ahead and erase this and move on to the next part. So next, what we want to talk about is how these macromolecules are formed, how these large molecular structures are formed. And to begin this discussion, let's talk about the difference between a polymer and a monomer. Polymers take small structures that are similar or identical in some cases and links them together, like so. The monomer is simply these small structures not held together. So these repeating units that haven't been put together yet are the monomers. Now, sometimes we will take these polymers and break them down to form monomers. So the reactions can go this way. We call this kind of a reaction hydrolysis. Alternatively, we can build the polymer, where we take these small monomers and link them together to form the polymer. We call this a dehydration reaction. Now let's spend a little more time talking about dehydration and hydrolysis. The hydrolysis reaction requires water to be put into the reaction, whereas the dehydration reaction releases water. If you think about something becoming dehydrated, they have less water. They're losing water. And so that might be an easy way to remember that dehydration reactions release water as they form a polymer. Hydrolysis, lysis, hydro for the water part, this simply means the water is being split. So water comes in, and as it comes in, it's split into two. Now sometimes these reactions can occur spontaneously, meaning just on their own, but often these reactions, both dehydration and hydrolysis, require a special molecule called an enzyme. We'll talk more about enzymes later, but what you should know about enzymes right now is that they speed up the reactions. Maybe this reaction could occur all by itself, spontaneously, but maybe it would take a really long time to achieve it. Enzymes make this occur faster. So, they speed up reactions. RxNS is just my abbreviation for reactions. And also, something else you should know about enzymes is that they can be proteins, and enzymes can also be RNA molecules. Okay, so let's quickly summarize what's on this slide here. First, these monomers, these small subunits, can be linked together to form a polymer, which is just a repeating series of these monomers. This polymer can be broken down through the process of hydrolysis, which requires water, into monomers. Alternatively, the monomers can be built up upon each other through a dehydration reaction where you form the polymer. And these dehydration reactions lose water. And lastly, remember, the enzymes, which can be proteins or RNA, help speed up these reactions. Alright, now that we understand the basic terminology and the basic parts of these large molecular structures, the macromolecules, let's begin talking about the macromolecules. Remember, we said macromolecules included carbohydrates, lipids, proteins, and nucleic acids. We're going to close this podcast with our discussion of carbohydrates. The next class will focus on the other three macromolecules: lipids, proteins, and nucleic acids. Carbohydrates we often think of as just simply sugars, but they can be more than just sugars. Turns out that they're very important in providing many different functions to living organisms. A couple of the main purposes of carbohydrates is that they provide energy. When we consume carbohydrates, say sugars, they are converted to energy within our cells. The other really important purpose of carbohydrates is that they are the building material of larger molecules. But to get started, let's think about the different types of carbohydrates there are. We can think of carbohydrates as falling into one of three categories: monosaccharides, disaccharides, and polysaccharides. So let's talk a little bit more about these monosaccharides. The first part, mono, means one, and then saccharide is sugar. So these contain one ring of sugars. Some examples of monosaccharides include glucose, fructose, ribose, and deoxyribose. Glucose and fructose are common sources of energy for a cell. Ribose and deoxyribose are often used as building blocks. Ribose for RNA, deoxyribose for DNA. Now let's talk about disaccharides. As the name implies, disaccharides contain two, di is two, saccharide sugar. They include two sugars. To form a disaccharide, you would take two monomers, so maybe you would take glucose plus fructose to create sucrose. So sucrose is an example of a disaccharide. So this would be an example of a dehydration reaction, where we take two monomers and we create a polymer. Other important disaccharides include lactose and maltose. Lactose is a common sugar found in milk, and maltose is a common sugar that's used in various industries, food industries. Lastly, I want to talk about polysaccharides. Poly means many, and sugars. So this would include sugars that contain three or more sugars linked together. I'm going to erase what we have here so we can talk a little bit more about polysaccharides. So this will be a discussion on polysaccharides. Polysaccharides are large polymers of glucose, typically. So you'll have several glucose molecules that are linked together to form this large string of glucoses, to form this polysaccharide. Now, I didn't mention this with the disaccharides, but I can mention it here. This bond that links monomers together, the monosaccharides, we call this bond here a glycosidic bond. Or you could say glycosidic linkage. I would accept either. Glycosidic because glyco for sugar, and then cidic for linking them together. Glycosidic. This is the same glycosidic bond that would be linking two monomers together, even though I didn't show it on the last slide. So I'm going to erase this so we can talk about some specific examples of polysaccharides. Alright, so now let's begin this discussion of the different types of polysaccharides. The first one I would talk about are starches. Starches are polymers of many glucose monomers. They are unique to plants, so only found in plants, and their function is to store energy. Remember, glucose is the main source of cellular energy. Glucose is broken down through the process of respiration, cellular respiration, to generate ATP, which is energy. So the plants can store this glucose in starches. Another point I should make out is that this polymer of all these glucoses connected together is usually unbranched. Now, animals don't have starches, but they also need a way to store energy. And they do that with this polysaccharide called glycogen. Like the starches, they are also polymers, large polymers, of many glucose molecules. Or we should say monomers. Unlike the starches, glycogen polysaccharides often contain branching. So we would say they're branched. These are found in animals, and like starches, they store energy. Because again, this glucose is the main source of cellular energy. Glycogen is typically found stored in the liver and muscles. I didn't mention it above, but starches are usually stored in the plants in these structures called plastids. Plastids include chloroplasts. So we will see large amounts of starches in the chloroplasts. Now, these first two polysaccharides that I talked about, their main purpose is to store energy. Now, there are two other polysaccharides whose main purpose is of structure and protection. So I'm going to erase these energy storing polysaccharides so I can talk about the structural polysaccharides. The first is cellulose. Like starches and glycogen, cellulose is also made up of, so it is a polymer of the glucose monomer. It provides structure and support, and it's found in the cell wall of plants. Because of the importance and the sheer numbers of plants found throughout the earth, cellulose turns out to be the most abundant organic material on Earth. Alright, this last one I want to talk about is called chitin. Chitin is similar to cellulose, so chitin also is a polymer of glucose molecules. Like cellulose, it also provides structure and support. It can be found in different kinds of organisms. It can be found in insects, and when it's found in insects, it is what the exoskeleton is made out of. That's the outer hard covering that we find on insects. We can also find it in fungi. In fungi, it makes up the cell wall. Fungi also have cell walls like plants, except fungi's cell wall is made up of chitin and not cellulose. Alright, that's all I have for you for this particular podcast. In this podcast, remember, we talked about the various components and terminology of large macromolecules, and then we went into an in-depth discussion of the first macromolecule, carbohydrates. For the next day, and in the next podcast, we'll finish this discussion talking about lipids, proteins, and nucleic acids. If you have any questions over this material, please don't hesitate to come see me, and I'll help you in any way I can. Alright, I'll see you in class. Bye.